Welcome to our first presentation today. If you missed my opening welcome message, you are at the Artisan Cheesemakers Winter Conference. Hopefully this is where you intend to be. If you did miss that opening little welcome message, I'll just let you know that I am Claire Natola, Education Coordinator for the Vermont Cheese Council. So I just want to remind you of a couple of logistical points. Please feel free to use the chat window to say hello, introduce yourself, or make any observations or comments to the group during our presentations. Feel free to test it now by just saying hello and letting us know who you are and where you're joining us from. But if you have questions for our presenters, please use the Q&A section to type those questions. We have Rachel Fritz-Schall monitoring the Q&A section for Bruno's presentation today. Thank you, Rachel. So just remember, put your questions for Bruno in the Q&A section, not in the chat window. If you have any technical problems, you'll want to email info@vtcheese.com. That's info@vtcheese.com, and I will put that into the chat window right now, so you have that information handy. And I'll remind you of that at the beginning of every presentation today. Okay. Don't forget that we will be recording each session, and we'll have those recordings available to watch later here on the SCED platforms, just in case you need to step away. So we will email you when the full slate of recordings is ready to watch. So let's get right to it. I'd like to introduce you to Bruno Martin. Bruno works at the INRAE Joint Research Institute on herbivores. His expertise is in cattle product quality, particularly milk and dairy products related to the husbandry systems and practices, as well as farm sustainability. He has over 20 years of experience in French and European research and development, which he will be talking to you about today. So, Bruno, welcome. Thank you for presenting for us today.
Okay, thank you very much for the introduction. Hello, everyone. I'm really very happy to give this presentation today, and I will start sharing my screen. Okay, so can you see my screen? It's okay? So I prepared this presentation with my colleague Céline Delbez. She is a researcher at INRAE as well, and she's working in another research unit called Joint Research Unit on Cheese. So both we are dealing with the link to terroir, and I am on the management of animal part, and Céline is dealing mostly with the cheese production and microbes in particular. So today, we would like to focus mainly on the latest on milk quality research for traditional cheese production and raw milk production more generally. So just a few words about our institute and where we are located. So here you have a map of France. You can see Paris with the next Olympics coming. And our research facilities are located really in the central part of France, in this part in which we have some small mountains. We are at between 800 and 1,000 meters elevation, and we have different facilities. So here you have a picture of our research facilities where I am currently. We have also an experimental farm. You have a picture here. This experimental farm, in this experimental farm, we have two breeds, Holstein cows that you most know, and Montbéliard cows. That they are red cows that maybe you discover here. We have also a cheese plant factory in which we are able to process small amount of milks. And one specific point for these research facilities is that we have in our mountains a huge biodiversity in the grasslands, which allows us to make some researches on this. So we will deal with the link to terroir. So what is terroir? More generally about the geographical indications. As you know, we have two different kind of geographical indications, protected designation of origin, PDO, and PGI. In fact, there are separate type of intellectual properties, and they are different from the IP we know all, relying on brands. Here, it's linked to a territory and to a human community living in this territory, what we call the terroir. And the definition of a PDO is that it is a product that originates from a territory and whose characteristics are linked mainly to geographical environment, including human and natural factors. So we have in France and in South Europe, let's say France, Italy, Greece mainly, Spain as well. We have quite a large number of different PDO cheeses. In France, we have 44 of them. So what is the terroir, in fact? There is no traduction. In fact, it's a French word, terroir, linked to the soil, in fact, something between the soil and the territory. And in fact, it's defined as a geographical limited area where a human community generates and accumulates along its history a set of cultural distinctive features, knowledges, practices based on a system of interaction between biophysical and human factors. Okay, that's not so easy. That's quite a long definition. Let's say that for the definition of terroir, in fact, we have some natural factors linked to the geographical area. We have some human factors, human organization, linked as well to the geographical area, but also there is an historical aspects, and both of them result, in fact, in original production system for the cheese and original practices. And the key question is there a link between this production system and practices we have locally and the products? Here we are talking about cheeses, and is it linked to typical products? So for animal products, let's say that the link to terroir, in fact, rely on soil and climate that gives some specific aspects to grasslands, to the diet of the animals. There are also specific animals that were developed in a specific territory, may give specific characteristics to milk or carcass, and finally to cheese and meat. So we are dealing here with direct or indirect flows of molecules and microbes that are very important. And within this general scheme, in fact, we have some regulation factors. So the soil and the climate, the local environment, in fact, is shaping the farming system and practices. Farmers may interfere on this with their practices on soil, on grasslands, just to define the diets, in the selection of specific animals, specific breeds that can have an impact, and more generally, the collective know-how related to animal production, but as well to cheese production and to process, may give specific cheese by the end. So in fact, in this scheme, we have two main aspects that we will deal with in this presentation. The first one is related to forages, specific forages and type of grasslands linked to the local environment. And the second one is related to specific breeds. So during this presentation, we will talk about forages, about animals, and about microbes. So for the first part, it is just a brief summary, because I think that my young colleague that presented last year made an in-depth presentation of our recent results related to this. So what can we say about grass and sensory properties of dairy products? If we are talking about milk, raw or pasteurized from cows grazing, well, we all know that they are very different from the flavor point of view from milks originating from cows fed corn silage or concentrate-rich diets with stronger flavor and a higher barn flavor in particular. we can appreciate or not. That's another question, but they are specific link. When we are talking about cheeses and butter from crows grazing, we know that they are more yellow and they have a really a more melting texture. And they have also some stronger flavors, even if for butter, they are in general considered as less rancid. So both for milk, cheese, and butter, in fact, we can say that there are products that are more appreciated by consumers in general and overall. We know much about the mechanism underlying these different relationships. Some years ago now, we have been focusing on the botanical diversity of pasture and cheese sensory properties, just because, in fact, the botanical diversity of grassland is really related to place, to a soil, to a specific climate, and so on. So it is really part of the terroir and of this link to terroir. So what we have seen and demonstrated is that cheeses from diversified grasslands in comparison to seeded monospecific grasslands or just with 2 or 3 different grass species, we have an increase of omega-3 fatty acids, which is quite positive for the consumers. We have as well some secondary metabolites that are transferred to cheese. So here you have a small illustration. And if we compare, just like a mirror, the grassland terpene composition. So terpenes are secondary metabolites from plants, from diversified dicotyledon plants in particular. You have 2 types of grasslands here. One that is very biodiversified, the other one that is rich in grasses. You can see that we have much more of these terpenes in this grassland, and that we can find them in the cheese. So there is a transfer from the plants eaten by the animals and directly to the cheese. So we saw that this transfer was associated with a slower ripening process and a higher aromatic richness of the cheeses. We do not know exactly if these secondary compounds are really involved in this association, but we think that they may be involved, linked to the ripening process, in fact, and it seems to slow down the ripening process and give, by the end, a higher aromatic richness within the cheese. So it has been seen with the Beaufort cheese, that is a hard-cooked cheese, and as well with the Cantal cheese that is coming from the area of France where I am come from. So we can say that at least there is an aromatic signatures of the grasslands into the cheeses. So that was just a very brief reminder on this. I would like to focus a little bit more on the animal. And when we are talking about animal, we are mainly talking about breeds. So here you have some examples of results that we had when we compared cheese made with the 2 breeds that we have in our experimental farm, Holstein on the one hand and Montbéliard on the other hand. So in fact, we know that the fat content of the Holstein cows is usually higher. And in this experiment, we chose to standardize the protein to fat ratio so that the fat in dry of the cheese was the same. Here we had slightly yellower cheese with the Holstein breed. And when we are talking about the sensory characteristics of the Saint-Nectaire cheese, you have a picture of this cheese here. We had a more melting texture with Montbéliard and more intense flavor as well. So you can see that these researchers are quite old. They were, in fact, the first one that we made on these aspects, and we had no real explanation of the differences that we had. More recently, we have confirmed with the Cantal cheeses, and we saw that the more intense flavor was partly linked to some enzymes that we can find in a higher amount in the milk of Montbéliard cows, and that the more melting texture was partly linked to the fatty acid composition of the milk, that is depending partly on the breed. If we are talking about more evident effect of the breed, here you have the Tarentaise cow. It's a very specific cow that we find in the Alpines, in the Alps and Alpine mountains. It is very well adapted to this environment. And we found some specific aspects related to one casein, so the beta casein with a specific variant that we find only in this breed. So this specific variant has a specific structure, and that leads to a slower breakdown of this casein during the ripening, leading to specific peptides, and they were responsible for the apparition of a pungent taste, which is not very positive if we are just talking like this. And as well, we have seen that the milk has a large micelle, a low calcium content, which gives, when it is a less firm curd, and that originate from of fat losses into the whey. So this decreases the yield and the texture. So you will say, okay, this is not very positive. Pungent taste, lower yield, less elastic texture. That's true, but the farmers decided nevertheless to try to keep this specific variant, because they said, okay, this is our specificity. And well, all the cows are not concerned with this, but they could be. And the last example that I wanted to give you is focus on the Salaire cow and Salaire cheese. So here you have a picture of this very specific cow that is that we have here in France, and the corresponding cheese. So the Salaire breed, in fact, is native from France, from the central part of France where I am. In fact, in the 70s, it was a dual-purpose cow for beef and for milk. Now it is mainly a suckler cow, but we have around 2,000 cows that are still milk. They are only a very low proportion of the total population, and the traditional milking of the Salaire cow is made with the calf. You can see that the production is quite low. So when it's a very specific system. So first the calf is allowed to suckle to induce the milk ejection. The calf is then removed to proceed with the machine milking, and finally the calf is allowed to suckle any milk remaining into the udder. So it's a long process. This milk is used to make a very specific cheese. It's a farmhouse hard raw milk cheese. When cows are fed only on pasture from April to November, and the cheese making is done in a wooden vat that we call in French the gerle. And the Salaire tradition is made only with the Salaire milk. So you have here a picture of this specific wooden vat. So the questions we had were the following. Are there specific characteristics of Salaire milk and cheese? Is it possible to milk the Salaire without the calf? And if yes, does it change milk or cheese characteristics? So what we our results are the following. In fact, we have seen that when the calf is suckling, we have a low milk fat content, because the fat content of the milk is really higher by the end of the milking. And this milk is drunk, in fact, by the calf. So it results in low cheese fat in dry matter and a texture that is quite firm and grainy and less melting, which is something that is really typical for this cheese. And here again, with this very specific breed, we have identified a specific variant for kappa casein in this case, and we have seen that this variant is responsible for a very low coagulation. And also we have seen that the microbial ecosystem was really richer when the calf is suckling before the milking. And we have also milks that have lower somatic cell counts, and we are wondering if these as specific aspects may explain why the ripening of this cheese is so slow, and why we have very soft, in fact, but very rich flavors for this cheese. So here we answered the different questions we had. So yes, there are specific characteristics of Salaire milk and cheese for casein, for coagulation properties, for microbes, for texture and flavor. It is possible also to milk the Salaire cows without the calf, but not all cows accept to be milked in a normal way, let's say. And when we do this, we lose some aspects of the typicality because the fat is increased and there is a global loss of typicality. So we can say that for Salaire, it's a slow cow. We have a slow cheese. And for this specific cheese, maybe slow is the key word for terroir of exception. So I gave you some examples of results that we had on the specific effect of animals. And now we will move to our most recent results that we have on microbes. And we deepened all these aspects, in fact, thanks to the development of new methodologies to assess the microbial composition of the milk and the different environment, in fact, that are the metagenomics, in fact. So, cheese microbiota and terroir. So, in the milk, we have some local microbiota, very important when we are talking about raw milk, of course. They are driven by practices, animal feed, milking, brine, equipment, and premises, and etc. Of course, we also add commercial starters that are inoculated into the milk. Then when we go to the cheese, of course, all these microbial communities are selected by the process, by specific aspects linked to water activity, to pH, to temperature, and so on. So we are selecting these bacteria. And in the cheese, it results in a specific cheese microbiota. So we know that this microbiota is really essential for the development of the sensory properties of the cheese, but in fact, little is known about their biodiversity and their specific drivers. And they are the recent researches we are making. So recently we developed a huge national program that is called MetaPDO cheeses, and they correspond in fact to an unprecedented sampling campaign in all the PDO cheeses that we have in France. So this program was coordinated by my colleague Céline Delbès and also Françoise Irlinger from INRAE too. So we collected, in fact, cow milk and cow cheeses goat, and ewe milk and ewe cheese as well, and Roquefort in particular. So what we have done here is that all the 7 cheese families that we have are represented. We go from blue vein cheeses to hard cooked cheeses here, Cantal or Comté or Beaufort cheese in particular. And we sampled the surface and the core of these cheeses. So we have 1,200 cheeses and almost 400 milks that were sampled and chosen to cover really the diversity in French regions, in season, farm size, feeding practices, milk processing, starter culture, and so on. So we have a majority of cow milk, of raw milk as well, and we have some farmhouse cheeses, but also corporate dairies that were involved in this project. So here we move to the results. We found a huge diversity of bacterial species and fungal species as well, as you can see. And this high diversity of indigenous microbial species in cheeses is largely exceeding the microbial cultures known to be added for cheese making. And what was quite expected is that, in fact, the PDO was an important driver for the microbiota of cheese after the cheese family. So we have first the cheese family, and within the cheese families, we have the different PDOs that can be separated, as you can see, according to the microbial ecosystem we can find into the cheeses. What is more maybe interesting and related to this famous link to terroir is the microbial composition of the milk. Because here we have no process able to select the different microbiotas. And we have seen here again a huge bacterial and fungal diversity that you can see here. And the dairy ruminant, in fact, is a major driver for milk microbiota composition. And here is the most interesting result, to my point of view, is that the PDO is the second most important driver for the bacterial microbiota of milk. That means that if we are comparing a milk coming from one part of France or a milk coming from another part of France, here you can see that we have a very different microbial composition. I was really surprised by these results. It means that the local specificities related to climate, to farming practices, and so on, is really a very important driver for this. And this is, of course, related to the terroir. So now, what are we doing, in fact, after these first results? We are trying to go deeper in the microbiome, and we are trying to decipher the transfer of the microbiota across the dairy system, starting from the soil, in fact, and until, of course, the cheese, and also the gut of the consumers, because we are expecting some differences in the gut microbial diversity and composition. So we are still working with raw milk cheeses, and we are dealing really with very complex microbiota, as we have seen. And we think that all these researches may be related to the One Health concept and also the One Quality, because specific environment, specific microbiota may have an impact on the gut microbiota of the consumers and maybe on their health. So the first results we have on this transfer of microorganism was published a few years ago. 5 years ago, in fact, and here we have represented the number of bacterial strains that are common to teat, to milk, to cheese core, and cheese rind. So, for example, you have all the different microbial species we have in milk, and the one that are common, for instance, with teats. Okay, so we have 9 bacterial species that are common to all the environment we have been dealing in this experiment. And we could conclude, in fact, that 85% of the bacterial species present in milk are also detected on teat skin. So it means that teat skin really is an important reservoir for bacterial diversity for milk and cheese. And 27% of the bacterial species present in cheese were also detected on teats, including the bacteria potentially involved in the flavor development. So other preparation before milking is maybe very important for the explanation of this bacterial diversity that we have in milk, and that is so important for the development of the cheese flavor in particular. So now the perspective.
30 minutes left.
Okay. So I will conclude this first part very briefly. So for this perspective here, now we try to understand the role of microbiota in the adaptation of dairy system to climate change in particular, and we are focusing on the drivers that may influence the microbial diversity and microbial transfer from all the components of the farms until the cheese and until the gut microbiota of the consumer. And we try to explore this in a wide range of dairy systems and geographical conditions. So stay tuned for the next result we will have on this. So just to conclude this first part, we can say that now the link to terroir is partly deciphered, but only partly, and that forage, animal and microbes are really key players that must be considered as a whole. In the first research, we have been working on forages and animals, and now microbes are really central for this explanation. And their determinants rely on local production system and practices that are driven by natural and human factors. So we are really in this complex relationships between natural factors, geographical area, human factors, and practices, and the link with the typicality of the products. So are there recent results? No, in fact, linking products to terroir, the goals were already talking about this, as you can see on this. And I would like to thank you for your attention. I'm open to any questions.
Well, hello. We got quite a few questions. So let's go ahead and we'll start at the start. First question is from Suzanne at Shepherd's Way. Is the increase in the terpenes in pasteurized milk as well as in raw milk?
Yes, we have no impact. We have checked this specific aspect, and we verified that it's both for raw milk and pasteurized milk. In other words, in fact, the thermal treatment has no effect on the terpenes that we can find in milk.
Next, is the fat and dry matter tested after molding the cheese or once the cheese has finished aging and is ready to be harvested?
Oh, fat and dry was tested just before the consumption by the panelists. So at the end, when it was harvested, yes.
Paul Lawler from Cherry Grove Farm asks, I assume we are talking about raw milk Saint Nectaire and Cantal, which we don't see over here very often.
Saint Nectaire raw milk, you don't see there very often? Yeah, of course. I know that it's very, very difficult to export any raw milk cheeses, and well, the aging for Saint Nectaire is only 4 weeks, I think. So I'm not sure in the US regulation it is allowed or not. Well, most of the works that we have been doing were made with raw milk cheeses, and but it's a very interesting question, because of course, when we are dealing with raw milk cheeses, we are considering all the last aspects that I presented, and we are not eliminating all the microbial biodiversity that we have into the milk. And when we made similar experiments comparing different breeds, different diets with pasteurized milk, in fact, we saw some relationships, in particular for texture, the influence on color. Of course, it is related to the carotene content of the milk, and really directly related to animal feeding. It is not influenced by the pasteurization. So texture, neither. Texture really relies on the fatty acid composition, and the fatty acid composition is not influenced by the pasteurization. But for flavor, in fact, the difference we have between the different diets are reduced when we are talking about pasteurized cheeses. And for us, it's quite important, because all the PDO that really rely on this link to terroir are not raw milk cheeses. Some of them are pasteurized cheeses, and well, we know that the link to terroir in this case is not completely disrupted, but at least it's not as strong as it is with raw milk.
Okay, we have several questions about teat treatment and udder cleaning. So first off, what are they cleaning the udders with after the calves finish feeding before they proceed with milking the Salaire cows?
In fact, there is no cleaning. No cleaning with the Salaire cows. Well, I've been talking about Salaire cows because they are really very typical, and I'm not sure. I know that there is another breed in the south of Italy that is the same. It's Modicana cows, and they do not want to give them milk if the calf is not here. So it's exactly the same. And the farmers, in this case, proceed with the calf. So the calf is first suckling and is cleaning the teats. Is cleaning the teats. But then when the calf is removed for the machine milking, the farmers do not clean anymore. So there is still some saliva as well on the teats, and we can find this saliva in the milk, and maybe it's also part of the very specific features we have with this specific cheese.
Regarding the Salaire milking, how much milk are they aiming to get from the cow before reintroducing the calf to suckle the remaining after the initial milking?
Well, in fact, the cow is deciding. The cow is deciding, because they try to milk as much as they can until there is milk available for the milking machine. And when there is no more, the calf is reintroduced, and the calf gets the remaining milk, because it is much more efficient to get the milk than the human. Than the human. So well, but it's a complex balance, in fact, because if they leave the the calf really free. And we try in some recent experiment because in France in particular and in Germany as well, we have some consumer or social expectations regarding the removing of the calves at birth. And it is less and less accepted, in fact, by the society, this specific practice. So we are trying to make some experiments in which we leave the calf with its mother for different durations and so on. And of course, if the calf is free, they will drink a lot of milk, up to 10 or 12 kilograms, in fact. So in the salaire production, the total production of the cow is not so high, and they have to solve a difficult balance between leaving enough milk for the calf and getting as much milk as they can for the cheese production, of course.
In France, are the animal varieties used throughout and in different parts of the country? Or does one part of the country tend to have one animal, goat, sheep, or cow? Are there some animals that are only in certain regions?
Okay. No, it's quite unbalanced according to the region. In fact, I don't know exactly if you know very well France, but the south part of France is a Mediterranean area. It's quite dry. And in this part, we have a lot of goats and ewes as well. So Roquefort cheese is really made in the south part of France. In the wet mountains, let's say in the central part of France and as well in the Alps, we have mainly cows. And of course in the western part of France, that is the part of France in which, well, let's say more than half of the milk produced is coming from. So it's quite unbalanced.
Are there cow-calf systems other than the salaire, so in other parts of France with different breeds?
Yeah, in fact, we made these experiments with the salaire. We have been starting working on this maybe 10 years ago, or something like that, little bit more. And in the experiment that we made, we compared salaire with their calves or not, and also Holstein with their calves or not. And we had a big audience with these results because there is a pressure, as I was saying, societal pressure indeed, for this specific practice of removing the calf at birth. And now we made some surveys and so on. We have more and more farmers starting to leave the calves with their mother. They are very limited. They are quite limited at present, but it's much more relevant in Switzerland or in Germany, where the attention for animal welfare is really higher than here, what we have in France. It is quite developing.
I think that most of the other questions really you hit on already in answering, and I'm not sure if you want to go ahead and move on to the second part of your presentation.
Okay, I can move. So I will share my screen again. So this is the new presentation. Okay, I will put it in presentation mode. So in the second part, just for a few minutes, I will be quite rapid on this. I would like to say some words about a very large European project that I am managing with one colleague, that is Cécile Berry, So it is the Intact project. So in this project, we are trying to develop innovative tools for the assessment and the authentication of chicken meat, beef, and dairy product qualities. So we are 21 partners across different European countries. We have 10 countries involved, and in the partners, we have a majority of academics, but also 3 industrial partners and 7 SMEs. And we are dealing with the one quality concept. So we are mostly focusing on the intrinsic quality of the milk and also the other products. Namely, we are dealing with safety, nutritional value, and sensory features, but also with the sustainability of the farms that we call the extrinsic quality, in fact. So the main objective is to have a wide view of the relationships that may exist between the diversity of the farming systems that we have across Europe and the one quality of the products. So that was asked by the politics, in fact, just to drive the subsidies and so on according to the different aims. So the challenges that we have is to develop models of the impact of farming system on quality trace, and to co-construct tools with the food chain actors for rapid and cost-effective prediction of the intrinsic quality of livestock products, also multi-criteria scoring tools, and by the end, the fourth challenge is to promote innovative husbandry practices, complying with high quality of products and sustainability, and the practices adapted in the most extensive and the most intensive farming system that we have across Europe. So very briefly, some words about the methodology that we have in this project. In fact, it's a multi-actor approach. So we try to co-construct with agri-food supply chain actors in order to give priorities to farming system and quality traits that we measure in this project, and to take into account consumers and citizen demand. We have a farm scale work for product sampling, for assessment of farm sustainability through surveys, and for implementation of innovative practices in different living labs. And for the scientific work, we are doing measurements, analytical measurements, of course, developing the tools, different tools. So here you have a sort of global view of the diversity of the farming systems that we have at the European scale. So we have, I think they are much smaller than what you have in US. For instance, we have in this part in Romania, very, very small farms. In fact, they are semi-subsistence system. We are not dealing with this because it's only a few animals within the farms. We are mostly in this project dealing with the other farming systems. So in Mediterranean area, in Italy, in South France, and also in Spain, Portugal, we have quite intensive indoor systems. You can see that the farms are larger in Spain with high yielding animals. The situation is quite different in Italy. We have smaller farms, but really indoor, no access to pasture, no access to outside, and no access to pasture. On the contrary, for example, in Ireland, we have smaller farms. You can see in average 60 hectares. That is not so much. Much extensive farming system that is relying mainly on the use of grass. In fact, they are quite extensive if we are talking about animal production per capita, but they are quite intensive if we are talking about nitrogen input on the grasslands, in fact. So this is another farming systems. In UK, we have in fact larger farms. And in Northern Europe, in particular in Northern Germany, we have the largest farms that we can find in Europe, with from, let's say, from 200 to 1,000 cows, or a little bit more. The average here is 300 cows per farm. With this classification, in fact, in France, here we have mainly two main farming systems. The first one in the western part of France, that is really the most common one. You can see 90 hectares, 75 cows per farm, and 8,000 kilograms per cow and per year. And in the mountainous region of central France and in the Alps as well, we have smaller farms with a lower milk production per capita and per year.
Bruno, 10 minutes remains.
Yeah, okay, thank you. Very nice. So it gives you really an overview of the diversity of the dairy farming systems that we have in Europe. So for our project, we are working with 80 farms across Europe, and they represent 18 farm groups that were chosen with the stakeholders, with the farmers, with the dairy representative. We are going from very extensive farms. Most of them are organic farmers. You can see, for instance, in Switzerland that we have these farms we were talking about, leaving the calves. So they have a specific label for this mother bond. No concentrate, only pasture and hay, and very low production, quite low production, and quite small farm as well. We have similar project farms also in France, but in this case it's not mother bond. So mother bond is really interesting for the stakeholders, because they asked us to work specifically on this. On the other side, we have very intensive farming systems. For instance, in North Germany, based on TMR, on corn and grass silage, and very high concentrate levels. They are quite large farm for Europe, and also in the western part of France, we have these quite intensive farms. And we have as well all the intermediary with a semi-extensive and semi-intensive farm. So we are trying to deal with all the diversity that exists at the European scale. So we sampled all these farms. Now we are making some analysis related to sensorial and technological traits, to nutritional quality of milk, and to safety of milk with the contaminants, dioxins, PCB, PFAS, heavy metals, mycotoxins, and so on, and much more positive aspects, relying on fatty acid composition, phospholipid, minerals, and vitamins, and so on. We are dealing as well with the environment, with the impacts through LCA assessments, and also services given by the different farm types, animal welfare, and farm economy as well. So thank you for your attention. The program is still running, and I would say stay tuned for upcoming results on this specific project. Thank you. Ready for some questions if you have.
Thank you so much, Bruno. Rachel, do we have additional questions?
So there's one question from the first presentation about the you referred to the PDO as the second most important factor, and this Christine is wondering what the first most important factor above PDO was.
For cheese, it was the cheese family. If we are dealing with blue vein cheeses or with hard cooked cheeses, of course, the microbiota of the cheese are very different. But within the cheese families, the PDO are the second one. And it was exactly the same for the milk microbial composition, which is more surprising, in fact, because I was thinking that the milk microbial composition was mainly relying, in fact, of hygiene practices before the milking. What type of disinfection are using the farmers and so on. And I was not expecting any differences according to the PDO areas and small PDO territories. And even if the farmers' practices, except when, of course, when we are talking about milking animal with calves, of course, here it's very different. But when we are not, the farmers' practices are not really related to the area where the milk is collected. And I was really surprised to see such big differences in the microbial composition of the milk according to these areas. And well, it's quite, in general, when we are talking about the microbial composition of the milk, we are talking about some species only that are analyzed by culture-based analysis. But when we are using these new generation techniques for the microbial analysis, we can see a huge biodiversity. I don't know if you have seen, but we have more than 1,000 different microbes that we can find in the milk. Wow, it's just tremendous. And well, of course, all these microbes are not involved. They are selected through the milk process, through the cheese process, of course. This is the main aim of the cheese process, but they are different and they can play a certain role for the development of the flavor that is so much non-understood, let's say. Let's say.
Let's see. There was a question about the if there was a universal methodology of pre and post milking teat cleaning or dip, and also just what the impact of cleaning has on the ability for the of what the teat can contribute to flavor.
Yeah, there is in the first results that we had at the French level, at Italian level as well, we were really disappointed with the low bacterial count that we have in France, at least. Very, very low bacterial count due to a very high hygiene that is developed within the farm with pre and post cleaning, with disinfection of the teats and so on. So we were saying, okay, we have so low amount of bacteria in the milk that can we still talk about raw milk? we have almost nothing in the milk. But in fact, after we were reassured about that, because even if there is quite a high process, high cleaning procedures, there is still some diversity on the teats, and they can further develop into the milk. Of course, when there is no issue with the farmers related to pathogenic strains and so on, Staphylococcus, Listeria, and so on, the advice for the farmer is to try to reduce the cleaning of the milks, and sometimes using some very specific procedures with with wooden
wood wool.
Yeah.
Wood wool. Yeah.
Yeah.
Sadly, I need to be the bad guy and stop the conversation there because we are out of time. I apologize. I think that we could go on for a much, much longer period of time. But I want to thank you, Bruno. This was such an informative presentation. And Rachel, thank you for fielding all of these fantastic questions from our attendees. So thank you both very, very much. We will reconvene in 15 minutes. That's 11:15 Eastern time, and we'll talk about recruitment and retention with an equity lens. So join me, and we'll see Christina Chang in 15 minutes. Thank you again.
Bye-bye. Thank you.